Hello and welcome to the Gene Therapy Insights podcast episode 2. Here at GTI, we step by step walk in the talk into the future of medicine, and the goal, of course, is to share the knowledge and spark novel ideas as we highlight not only the advances in the clinical stage gene therapies, but also the novel approaches which could be in non-clinical stages of gene therapy developments. And today is this kind of day when we will discuss nothing less than an extraordinary way of approaching human evolution. Call it controversial or call it groundbreaking, but keep the spirit of curiosity and the budding possibilities which may come to life. Today, our guest is Dr. Shahaf Peleg. Dr. Shahaf Peleg received the bachelor degree in biology from Ben-Gurion University in Israel. He received a PhD at the University of Göttingen in Germany. As a postdoc at Ludwig Maximilian University in München, he studied connection between the epigenetics and metabolism. He then started his lab at the Research Institute for Farm Animal Biology. Then himself, with a co-founder, Andrew Wojtowicz, they co-founded Luminova Biotech, which utilizes their research in optogenetic rejuvenation of mitochondrial membrane potential via harnessing the energy of light and converting it into mitochondrial chemical energy, which has a goal of translating this technology into a human gene therapy. I bet you've only heard of converting the energy of light into chemical energy in plants, and trust me, I did too. So today we will learn about this revolutionary and groundbreaking idea of taking a light-activating proton pump from a fungi and inserting it into a mitochondria of an animal to partially replace its energy source. We will discuss the connection between epigenetics and metabolism and hypothesize the experimental design of the studies to come, which will take this technology into a human clinical trial for diseases of aging. Expect to be enlightened by our conversation. Without further ado, Shahaf Peleg. All right, Shahaf Peleg, welcome to the Gene Therapy Insights podcast. It's a pleasure to have you on.
Thanks for inviting me. I'm looking forward for our little chat.
So today our conversation I'm very much looking forward to. It is such an exciting topic, and it is quite revolutionary in the way we can think of, again, billions of years of metabolism, how we can rethink this whole concept of, again, life and energy and implement it to human evolution. I know it's quite futuristic, but I'm so excited to learn about your work and your perspectives. So let's jump in with the first question. Tell us the story. How did you first got interested about, again, energy, metabolism, and the specific approach that you're using? Tell us a little bit more about that.
Okay, I'll try to tell the background story a bit. So, by training, I'm actually a neuroscientist. I study neuroscience in my PhD in Göttingen, where actually my starting point was epigenetics. So, histone modifications and transcription regulation, especially in the context of aging and neurodegeneration, is how I started. Towards the end of my PhD, I was really more interested in metabolism because the field really went through this direction that metabolism and epigenetic are tightly connected. Some really nice work by Katie Vellen showing acetyl-CoA metabolism support histone acetylation. So I gradually became more interested in metabolism, and that was the main focal point of my postdoc. That was how metabolism and epigenetic connectivity is regulated in aging. And yeah, so when I started my lab and finally I had a bit more freedom, I thought like, it's very nice to continue going in this direction, but I want also to have a bit out of the box, so-called crazy ideas. And for a couple of reasons we can discuss later, I was looking for a way to, let's say, not trick metabolism, but in a way replace metabolism. Because at some point I thought that, due to various work by other people and also myself, I thought that as we get older, to a certain point, like really middle age, we have actually an increase of metabolic activity. It's counterintuitive, maybe thought, and I thought that was bad. And I thought, is there a way to make metabolism work less hard and achieve the same results? And that's why I started to be interested in other ideas, which we're going to talk about today. I guess a lot about this energy light replacement. Yeah, and specifically about the light metabolism, actually, so the credit goes to another person called Andrew Wojtowicz from University of Rochester. He invented the tool that we're going to talk today. We are partners. And his student Brandon Berry that spearheaded this project. And at that time, I was thinking about light energy conversion in mitochondria. There were papers showing that you can use light specifically in proton pumps that can generate proton gradient like in our mitochondria using light. And I was thinking how to use it, where Andrew and Brandon paper actually got published in 2020 or 21. And funny enough, I saw it on Twitter and I fell in love with the concept immediately. The next day I was already writing Andrew on an email, we must collaborate. And this is where the story begins.
This is super exciting. So how did you come about in your thinking when thinking about metabolism, when thinking about mitochondria? And maybe you could give us just a little bit of sort of like overview and introduction as a friendly reminder about the metabolism, the basics of metabolism, how this mechanism evolved on Earth throughout different organisms, from bacteria, fungi, plants. How are we different as mammals, as humans, and how could you even re-envision utilizing something that came from more archaic form of life and utilizing it into human?
Well, that's really complex and multi-layered question, I would say. So where to start? So from the evolutionary perspective, we think our metabolism is very beautiful. We think at some point in evolution, our very early ancestors managed to incorporate mitochondria and elaborate our metabolism to extent that we can generate more ATP. This is the metabolic coin of life, if you want to call it like that. And around long time ago, I would say more than 1.5 billion years ago, we have an evidence that there was an increase of oxygen in the atmosphere, and that animals, very early animals, were able to use it with the mitochondria to generate more ATP. And since then, unfortunately, our mitochondria or central metabolism of using food we acquire and oxygen to generate ATP, it became very much fixed. And I like always to provocatively say our metabolism is old-fashioned, because for basically 1.5 billion years, it hasn't changed. The elementary idea did not change so much. And we're still using it today, and it's perfectly fine to make us who we are. But we, of course, dealing with new challenge of aging. And I think our old-fashioned metabolism is probably not sufficient to answer metabolic problems that arise in aging. Now, back to my little part in this story, I came, as I said, from epigenetics perspective, and I noticed that during early aging, we see increase in histone acetylation. It's an epigenetic marker believed to activate transcription. And this is very much supported by acetyl-CoA. It's one of the key metabolites of the cell that is also is involved just before the TCA cycle that ultimately generate the ATP in the mitochondria. And I also noticed that in middle age, this acetyl-CoA is increased. And also some metabolites that are connected to ATP production are also increased. And I thought that's very strange, but among other things, it seems to me it's like walking in a hyperdrive, if you want to call it, to compensate for something. So my hypothesis was that as we get older, the body tries to meet the energy demands by walking a bit faster or putting more energy into that. And you can think about it like driving a car. If you drive a car on a flat road in 100 kilometers per hour, you press the gas to a certain extent. But if you go up the hill, you need to achieve the same speed, you need to press the gas a bit stronger. And by doing so, you have the same speed, but you put more stress on the car, engine is getting overheated. So I was thinking from a metabolic perspective, what happens when we go up the hill, and the metaphor is start the aging process, if we have some imaginary horses or imaginary very strong people behind just pushing the car so we can press the gas as if it's 80 kilometers per hour, but the extra push gives the 20 extra. So we achieve the same speed or the same energy demand, but putting less stress on the system. So that's brought me to this idea of using energy replacement. Why not use solar energy to generate the same amount of extra energy, but doing it in a cleaner way? So this is in a bit try to answer this very complex question.
No, this is definitely extremely fascinating, and I think just how much we know already, but just by studying biochemistry, and this is something very fundamental that is being taught to students in the biochemistry courses, but the process itself is just so incredible and fascinating and so complex that I see in just the field of the science of aging, in the field of science of different disorders, how mitochondria is often in the center of everything. Of course, there are disorders that, affect other parts of the organelles of the cell, or different mechanisms of action of how disease is manifested, how things may go wrong inside of an organism. But ultimately, this energy system is just at the center of everything, the sustaining life within the cell. And it's so important, not just for energy production, right? But it has a lot of other functions, including some signaling functions to tell the cell when to go through a programmed cell death or apoptosis. So when you think of that, and when you think about the clean energy source, like you said, how do you envision this to impact the mitochondria and mitochondrial dynamics within the cell? And just to step back, sort of like as a reminder that mitochondria is the most efficient in producing energy. It takes up the substrates that went through glycolysis and the other nutrients like amino acids or fatty acids, and it utilizes it within the mitochondria through the TCA or Krebs cycle in order to, again, produce some ATP, but as well as through oxidative phosphorylation that also occurs through passing of the electrons and creating that proton gradient, which is, of course, relevant to the mechanism that you use in the proton pump, that you are inserting as an extra source of protons that feeds into the ATP synthase in order to create more energy. So, to put this all into perspective, mitochondria can create some of the reactive oxygen species throughout this whole process I just described very briefly. And it can be damaging, but it could also be useful. Like in biology, we quite often don't see black and white, right? A lot of processes, even in inflammation processes, they're useful to a certain extent, and then they are deleterious to a certain extent. So if you utilizing this clean energy source, you insert in this pump, which I would love you to describe it just a little bit better, and where it comes from and what exactly does it do. Like what happens to the rest of the mitochondrial dynamics? Are they still, the ROS, the reactive oxygen species, are they still able to signal that cell needs to go through a clean cell death versus not, which could also be damaging to aging, when the cell becomes senescent or anything of that sort. Would you go into that in a little bit more depth, please?
Yeah, I think it's very interesting what you just mentioned. So this concept of cleaner energy and black and white, so how it affects ROS production, how it affects mitochondrial dynamics and structure. So the simple answer is we don't know yet. This technology that we have been utilizing is very new, and we know very little about what really happens. Andrew and Brandon showed this using this MtON, and I'll touch briefly how they done it. That the MtON tool can generate ATP with shining light without the substrate that you described that usually are produced by the TCA cycle. So we know the light can generate ATP. It seems when you do add substrates, you can generate the same amount of ATP, but with less oxygen. This is the last step in the electron transport chain. So when we talk now, we constantly breathing. How essential this process is. So if we stop breathing, we will die. But these two actually enable you to make ATP without the need of oxygen, which is nothing less but revolutionary in my mind. So bringing back to the topic, it seems as if we can hypothesize that if we use MtON and the right light in a sufficient manner, we would be able to generate enough ATP without producing as many ROS, because there is less potentially utilization of oxygen. But we don't know what happens to the other parameters, the metabolic flux, the fission of mitochondria and so on. That's something that definitely need to be tested. Now about the MtON itself, so again, I cannot stress it enough that Andrew and Brandon are behind this. I tagged along later for the aging perspective. So this light-activated proton pump is originated from fungi, an organism that we separated from them over 1.5 billion years ago. And Andrew and Brandon engineered this light-activated proton pump from fungi. They manipulated the sequence and they altered it in a way that it finds a new home inside animal. They did it in worms. So now this light-activated proton pump called MtON finds its way inside the mitochondria, into the inner membrane of the mitochondria, where when you shine light, it partially replaces this very canonical electron transport chain. So it kind of does similarly in the way that it generates the proton multiforce, so the PMF, and the light itself managed to move the protons without the need of upstream substrates. So this is a bit about the history. I came later because I was interested, as I mentioned, in aging, and we showed that indeed if you use this trick in C. elegans, they are long-lived. So this is one thing. Another point to say that already 30 years ago, another group did something similar, but in yeast, and they showed, for instance, that if you use light-activated proton pump, another one, and you put it in yeast, the yeast needs to utilize less glucose, suggesting that really this is the direction that the MtON can generate energy and replacing or transduce the energy in a way that we don't need food from or as much, which is of course now immediately come to mind, oh, it's like dietary restriction, do we need to eat less?
Exactly what I wanted to
also
ask
eating
you.
less, caloric restriction. Yeah, so caloric restriction, even Will Mayer from Boston, he showed it also affects mitochondrial dynamics. We actually we will be very curious to see if you can have some sort of additive effect, so we can have maybe more extreme caloric intervention or dietary restriction and use the MtON and somehow benefit from both worlds. This is something that really needs to be further investigated. Now, this whole concept of, you said like yourself, metabolism is very complex. So starting from glucose, for instance, there are a lot of biochemical reactions that a lot of our audience that study biochemistry in bachelor degree had to memorize all these enzymes and intermediates. And I think one of the intuitive ideas that when you have so many reactions, biochemical reactions, you will generate too much toxic byproducts, such as AGEs, such as lipofuscin, such as ROS. And while it's not black and white, I think my intuition would say that when we get older and maybe our metabolism is a bit in hyperdrive in middle age, there is less in black and white and it's more black. So there is maybe an accumulation of these toxic byproducts, which I think you can see, for instance, in the eye, highly rich with mitochondria, and during aging you have a lot of lipofuscin there that the cell cannot evacuate. Another thing which I just want to touch, this is good for, I think, what I said for middle age, but as we reach older age, like really old, 80 or 90, I think that the MtON will have a different advantage. In that case, the metabolism might be already impacted, that the mitochondria are highly dysfunctional, and therefore we might have a car that cannot, no matter how much you press the gas, cannot generate this speed anymore. It doesn't meet, so the old cell cannot meet the energy demands. And I think this would be like recharging the battery, where the MtON will be able to supplement the missing ATP. So this is another advantage. This is also an hypothesis we need to check by introducing MtON treatment at different stages in life, for instance. But this is still something that needs to be done.
Yes, this is super interesting. So I want to again put it in the perspective for our audience. So your research primarily is targeting the idea of aging and all of the age-related changes that show up molecularly, but at the same time they show up as the major diseases of aging, which of course could be age-related dementias, macular degeneration, cancers, I imagine, cardiovascular disease, right? So all of these diseases actually have, from my knowledge, both Alzheimer's, Parkinson's, and cardiovascular cardiomyopathies, myopathies, they have some of the markers of the changes in the mitochondrial membrane potential and changes of the mitochondrial function. It fits with the idea that by targeting the mitochondria, by helping the mitochondria to be healthy and to provide basically better energy sources, to provide the energy that the cells need, the cells will survive better. So this is I guess the simplistic way of viewing this approach, of even understanding of where this idea comes from, right? But of course, this could also be quite applicable to some other mitochondrial disorders, perhaps, whether it's a nuclear DNA gene mutation or a mitochondrial DNA gene mutation, if it impacts the function of the mitochondria and shows up early in life, where it could go anywhere from fatigability to early in life epilepsies, epileptic seizures, a lot of myopathies that show up as mitochondrial disorders, movement disorders. So with all of that in perspective, again, just to highlight how if this, comes to the point to be in humans, how much applicability this can have.
So I think you said it before, when you go to an aging conference, a lot of the talks revolve again around mitochondrial dysfunction. And mitochondria is a center suspect behind a lot of processes in aging and also related diseases. So I think if a disease or aging phenomena revolves around a dysfunctional mitochondria, I think the MtON, for instance, or the concept of light-induced energy replacement or enhancement is probably very attractive to test. Like you mentioned in AMD, for instance, the eye has a lot of mitochondria, as you mentioned before. It's very metabolically active. For instance, combining with cardiovascular diseases or diabetes, where you have a state of hypoxia, so less oxygen that reach these neurons and cause neuronal death. Of course, the MtON being that it can make ATP without oxygen, so mimicking hypoxic state, would be very attractive and straightforward to be tested. As you also alluded, cardiovascular diseases, here we go into the challenge of the obvious that any person in the audience would think, well, how you're going to get the light inside inner organs, which you just mentioned, and benefit from this technology. And I think this is a major challenge that we are now facing, and we are working on that. And I hope in the future we can also bring forward an idea how to tackle this.
So with that idea, again, the link between the changes in the mitochondrial biogenesis and the mitochondrial dynamics with age and with other disorders, whether they're mitochondrial or other type of disorders, and bringing it back to aging. You've mentioned that there is this intimate connection between the mitochondrial state and acetyl-CoA increase in middle age with increase in acetylation. And by no means I'm an expert in epigenetics, and not even close, but very curious also for our audience to give that perspective that there is some groundbreaking work right now as presented by David Sinclair in the field, right, that is targeting epigenetic reprogramming. And I know that they did it both in mice and now in non-human primates, where they insert, using an AV vector, they use it as a gene therapy to have a transient expression of 3 out of 4 Yamanaka factors, the transcription factors that are reprogramming the DNA inside of the eye where it was inserted, to kind of like give the information, to change the information to, like he says, more youthful type of information where the cell seems to remember which genes to express and how to act in response to whether it's environmental stressors that induce aging. But that ultimate result of that work was that the vision loss was reversed to a certain extent, and I think it was replicated in non-human primates as well. I guess he wants to take it to an idea where this cocktail can just be used, again, just as a cocktail without a gene therapy, something as a very simple approach. So where does it stand next to an approach like this, where you have to insert genetically a proton pump to mitochondria? Where do you put these two approaches in a big picture of how to tackle aging and age-related disorders and other disorders?
Yeah, well, that's really interesting point to discuss. Where to start? So I think what David and others have been doing is very, very interesting. It's very interesting idea to pursue. I think in the field of aging, you need to be mindful that if you ask epigenetic and aging, you ask 10 scientists what happened to epigenetic and aging, you get at least 11 to 20 different opinions. And I think it's worthwhile that, we try new things, and I really admire that David really go out of the box. This OKS treatment in mice is really interesting, and I'm curious to see how it develops. I'm a big fan of gene therapy, and maybe to go back a bit, there is a very beautiful piece that was published last year in Nature Aging by Yang Vig, and it has a very interesting concept. It brings forth the notion that the things that has been done now in anti-aging and longevity are kind of have very limited potential. So he doesn't think that what we do now, especially I guess in exercise, diet, and drug intervention, that can really make anyone live 120, despite what you see on Twitter and conferences. He really said there is a hard limit of human life. And he says, although in nature, other organisms, other animals like the Greenland shark can live 300 years, or turtles, he says really if we want to use advantage of this evolutionary concept that allow organisms to live that long, we essentially need to change something very basic. We need to change radically how the way our biology is being used at the moment, how our metabolism is working at the moment. We need to change it radically. And I really feel very much high sympathy and with what Jan suggests, and I think this is exactly the point. In order really, if you want to achieve increased health lifespan, you probably need to do diets, be happy, drink a bit, party a bit, use some supplements. But if you even dream, if it's even possible, I'm not sure if it's possible, but if you dream to live 150 years, you need something to change really radically in you. And the question is now, if OKS is radical, I think David think it is, and I'm very curious to see how it develops. In a way, you try to rejuvenate the cell, reprogram them to become young again, and be reprogrammed so they remember their young epigenetic state. And I think this is a very interesting idea, and I think it also alludes to the fact that I also believe that one of the only possibilities that I see, at least, to radically change our biology and biochemistry is through gene therapy. And of course it has also limitations, how our immune system is going to react to that. So when David used, and again, I shouldn't talk about David too much, he is the one that can talk about it, but using AAV in the eye is something that is gaining popularity. It has immune privilege, so you can use AAV a bit more with gene therapy and less immune response. The question is what happens when you do it in other organs, and so on, how to use the AAVs and other methods of gene therapy. But I think, for me at least, if we really want to dream about humans living 150, we need to go something very radical like gene therapy, change our blueprint that made us who we are. And I think the MtON is in a way radical gene therapy. That is, if it's going to work out, I think a lot of people would like to use. It changed radically how our biochemistry works. It enabled us to use the energy of light, something that none of our ancestors could do over, as I said before, this astounding 1.5 billion years. So you have your plant behind you, it can do this, but we cannot. But if we can, maybe it will give us another ability. Maybe it can combine with other technologies like OKS. Again, OKS is a topic for another podcast. A lot of people are also vocal about that it can cause cancer. Although you remove the fourth Yamanaka element, maybe it can cause cancer, and so on. The question also, one can think how the MtON can be disadvantageous if there is any side effects, and that's something that we also need to learn. Is using too much light can cause too much PMF that will turn us into
Of
vampires
course.
that we need to hide from the light because it's too much. That's a lot of things that we need to test.
So I want to definitely get a little bit more into this exploration of what it could look like. And just to reinstate, I'm all about futuristic vision and futuristic thinking and taking the small steps that are applicable, feasible, and getting us closer every day to testing those hypotheses that could bring us to a groundbreaking revolution. And just like we've, as humans, existed for like thousands of years without knowing that the mighty bacteria have the CRISPR-Cas9 proteins, or Cas9 proteins that were then called CRISPR. And now we're using them, again, to deliver gene therapies into humans. This just happened over the past 10 years, truly, thanks to the groundbreaking work of incredible people who found this technology and thought about applicability and thought about how this could be used in humans and beyond. So I definitely see how this, as a technology that comes from fungi, it's a light-activated proton pump, could be used in order to maybe alter us as species. And certainly it's exciting to even think and dream about it and what it could do. So in order to go a little bit more into depth, describe to us, how do you envision, I know you've done experiments on worms, C. elegans, that when you insert it, which is of course much simpler than a human organism, it only has such a, I don't remember how many, 108 nerve cells or something like that. It's just such a simple organism. I also believe you can shine the light through this whole body, right? I believe that it's translucent and probably just light can penetrate through the whole body. So this was a really nice confirmation of the principle of how this could cultivate more energy inside of the worm's body and ultimately led to life extension at a certain intensity of life that was shining on the worms. So how do you take this into, again, your work on mice now, right? It's more complex, it's not translucent, of course it has a more complex nervous system and metabolic system. And then how do you envision this to be taken into and translated into humans?
Yeah, I think that's a very important question to also discuss. So, actually, again, it was Andrew and Brandon that first tested it in C. elegans in worms, where it's much easier to shine light. In fact, actually, Andrew and Brandon, with collaboration with Minzo Kim's lab, they also showed this proof of principle that you can do it also in mammalian cells, so T cells of mice or HEK cells, oh, HeLa cells, sorry, I think it was HeLa cells. And they showed you can also use mammalian setup, at least in cellular level, cell level, to use this MtON with light to generate ATP. My role is more to think now how to translate into mammalian cells, not mammalian cells, mammalian organism, which you, I think, identified also the problem that whereas worm is simple and very rather flat, we humans, mice, we are 3D animal, we have a lot of barriers, how the light will penetrate inside. So first of all, in my lab, we started mice work, funded actually by Impetus Grant, and maybe this is an opportunity to shout out like this amazing Impetus Grant and also Evolution to extend, the people there actually try to fund some crazy out of the box idea. I think that they really recognize the need to promote out of the box project, but also I think I really like it. Like you mentioned with the CAS, it comes from a completely different field and it's implemented now in human health. So Impetus Evolution, I think, also recognize the idea of if you're not working in the aging field, but you have a great idea, come and apply, because maybe your idea is brilliant and nobody in the aging field thought about it. So we are funded by Impetus Grant and Evolution on that. When we are trying now to see more about, I don't want to go too much into that, but we go into the skin and see if MtON can impact some aging phenomena in the skin. Obviously, the skin is also like the eye, an organ that it's easy to shine light. Now, so me and Andrew, with another investor called Peter, the three of us, we started a company called Luminova Biotech, which is exactly now to try to promote the idea of translating these results to mammalian and human health. So obviously the two things we focus on, as I mentioned in the lab, the skin, but the company will start, or we are starting already experiments with the eye. And we also try to think how we can use it in inner organs. I'm not going to give any satisfactory answer. I'm only going to say that we have a very clear idea how to do this, and we are working on that. It will be a technology upgrade which hopefully will allow us to address it in organs. So to summarize this point, so we know it goes into mammalian mitochondria. We know in two organisms their cells are responsive to the light. And so now the question is just to test it on an organ like the eye and the skin and see how we can deliver it inside. There are a couple of ideas that people can immediately think of. We have a very clear idea, and I think with a bit of extra funding we can really implement it and give yes or no if it works.
That's right. So let's think about this a little bit further. So if we're targeting this in the eye is a very highly active metabolic organ. Vision itself takes up so much energy and has high energy requirements. Eye is also very complex and very compartmentalized, which is why it has been also in a way an easy target for inserting a gene therapy, where you can just inject very small bits of virus that either delivers a gene of interest or destroys a gene of interest, and it can be targeted specifically, injected specifically in the compartment of the eye. But the idea here, or the question here that I have, considering that different cells inside of the eye, whether it's retinal ganglion cells, the cones and rods, they have different energy requirements and they have different amounts of mitochondria. And the mitochondria also, they constantly change in those, right? From my memory, that there's constant recycling because of how much the mitochondria is used. So they're very, very dynamic. Pick up from that place and tell us, how would you imagine to inject a gene therapy in the eye that delivers the proton pump? Where do you expect it to go, and which light at which intensity would you expect to shine, considering that we're constantly exposed to light as humans, and now not even during the day, during the normal circadian rhythm and cycles, but during night where people are staring at their phones and screens. How do you envision that?
Yeah, I think this is a very beautiful point to discuss, and I'm unfortunately going to give a very unsatisfactory answer. We don't know yet. We really need to do a lot of testing here and a lot of optimization steps. How to inject which cells, which dosage. We really don't know, so I cannot really, it would be anything data-free observation, which is not good at this step. Regarding the light, it's also a bit of an issue because how you deliver the light during the night, where actually the eye is very highly metabolically active. Just a provoking thought, do we really, when we close the eyes, and I will close the eye now, okay? Is it really dark or I still see some light? I still see some light. The question is it enough? There's a lot of questions that need to be answered, but I'm very confident that this will work. And the idea is that, maybe I forgot to mention before, the idea is when you insert the MtON, the ratio between ADP and ATP is highly controlled on a cellular level. So the cell knows to regulate very well what is the ratio between ADP and ATP. And just by the fact that we add MtON to generate ATP, I think the cell will just readjust itself to go to the equilibrium of ADP to ATP level ratio. So again, as I mentioned, it will just be easier to do it, either because you don't need so much upstream reactions, or in old aging that you can go back to the youthful energy state. I think despite of the light and how many copies of MtON get which cell, this needs to be tweaked out. So I think this is a major challenge, not just for MtON, but in general in therapy in the eye. This is something we are learning on the way now as we start the experiments, and I hope in a year or two I have more to share, like more concrete question, how, which cells, how did we do it, and so on. I think it's just a bit too early to really go in depth, unfortunately, to this point.
That makes sense. So remember you mentioning that right now MtON is activated by the orange wavelength of light, right? And of course, my understanding is the orange wavelength is also present in the white light spectrum. So if we were as humans to be exposed to white light during the day, it would be activated, so to say. So is there a way to have a controlled stimulation where you could re-engineer this protein to be responsive to a particular wavelength of light that is somehow that we're not as exposed to? Like, can you elaborate on that?
Yeah, I think that's a very important question for technology upgrade. I think this is a question that should be addressed to Andrew Wojtowicz that, as I said, developed this tool. I think it will be interesting to try to either change the light activated proton pump in a way that it can also respond, for instance, for red light, because just red light can penetrate a bit more deep. As far as I understand, actually, the ATR is all transretinal. It's a cofactor that is needed for the activity of the light activated proton pump. And this is actually the cofactor that is responsive to the light. So I'm not sure how you exactly will change the light activated proton pump to be responsive to red light so easily. This is not really directly my field. However, I attended a conference a year ago and someone suggested maybe another light activated proton pump of organisms that live more deeper in the sea but still can use light. And maybe their light activated proton pumps would be more responsive to red light. So this is, I think, again, some exploration that can be very beautiful, and I hope someone is interested to collaborate with us and see if we can do it, if we can respond to different light wavelengths that can maybe be more beneficial in certain situation, like penetrate more, or if you want to use different light specifically in your home, so you can have a specific wavelength. At the moment, it is orange around 580, 590. Maybe we envision in the future that if this therapy is, need to be chronic, for instance, you can have at home specifically orange light while you do your normal day-to-day activities, watching something or eating, but you have the light that constantly help you make ATP in your eye and sort of attenuate its aging, for instance. So chronic but rather passive treatment.
Yes, that's quite interesting to think about. And I know that even nowadays it's not similar, not dissimilar technology, but it's also a way to stimulate mitochondrial biogenesis is via the photobiomodulation, where the mitochondria itself has already cytochrome C inside of it, which responds to specific wavelength of light, which is near infrared light. And it can also cultivate the mitochondrial biogenesis, help the energy within mitochondria, and it's already used through different devices by shining, again, the infrared light via helmets, which can penetrate, as you mentioned earlier, a little bit deeper into the prefrontal cortex, into the cortical areas, as well as it's used for the eyes and has quite promising results for some of the dementias and Alzheimer's and some of the eye disorders of aging. So I can definitely see how your technology can also be applied in the future with us using the light as a source of therapy, truly.
Yeah, the red light is very interesting because when we published our story, I got several emails about red light. But I think it's probably a completely different idea and approach. I dare say, while looking at red lights and especially maybe sunset makes us very calm and more happy, I dare say that staring at red lights without any change really in our basic biochemistry will really meaningfully extend lifespan. Definitely not the goals that we set ourselves to break this glass ceiling of living over 120. It's very different here because what we do with the MtON, we able to harness the energy of light and transduce it to a chemical energy with this new feature in the mitochondria. I think it's different, but I think, as you allude, if people use now red light at home or at spas or I don't know where, why not doing it with orange light that actually can activate something new? So I think it is possible.
So tell us, what are your, the most proximal future goals at Luminova? What are the biggest questions that need to be answered in the next few years? And what are your visions for the future to conclude our conversation?
I guess a lot of people advise is to focus on one thing. As a scientist, it's very hard to really do that. I mean, Luminova has, I think, 3 main goals. So one of them is the technology upgrade. So we expect in the next few years to be able to activate MtON inside a mouse liver or heart and muscles to show proof of principle that you can really use this technology throughout your body. I think it will be also much more attractive to people to invest or to think about the promise that you can target any cell in the body, whether just the skin on the eye. Although saying that, imagine how many people invest money and time in looking young or having good eyesight. But still, I mean, we want to really fully harness the potential of MtON, and that can only be achieved by doing it inside also inner organs. The second thing would be really we focus on the eye and the skin. We want to see if a proof of principle, the MtON will attenuate the progression of aging, if it can use as preventative approach. So you can use it when you are before middle age and generate less lifelong accumulation of toxicity and toxic waste, and by therefore you prevent a lot of prevalence of diseases. And if it can be used in older individuals that already suffer from partial vision loss or more advanced vision loss, and imagine you can use the MtON technology and restore your vision again. I cannot imagine what it means to someone that actually start losing their eyesight to be able to see again, just because now they can generate ATP that they could not use before, like in diabetic retinopathy when you have not enough oxygen. So this is another goal of Luminova, is to really see if MtON therapy can work in mammalian setting. We first now do the preclinical trials, and hopefully in the next couple of years we can already start the clinical trials in humans. And I've been working in the field of aging for 17 years, and by far MtON, and it's not commercial, it's really what I feel, it's by far the thing that I felt the most excited, I still feel the most excited in terms of, okay, we really have a chance to change something in anti-aging and longevity. It's so crazy, but it so much makes sense that it holds great promise, and I'm very excited to see what comes out of it, with it. And I hope in the next few years Luminova can answer these things. We'll see. I mean, if it works, I think, I dare say, a lot of people, it's hard for me, people ask me how many patients, how many people will use it, but why not everyone will use it? As long as you have the drive and the desire to live long and healthy, and you have something that you just put the eye drop, and you don't need to do anything else except from living your own life and being exposed to lights that you anyway expose, but this will promise you, generate you, that you will live healthy and longer. I think everyone that has this desire will do that. So there is enormous potential. And if this idea doesn't work, certainly I want to use this platform and say, I think we need more of this out-of-the-box crazy ideas. It's the only way to move forward. And people should stop asking, what is the consumers, who is going to use it? Everyone that is interested to live longer and healthier. And I think a lot more people really wanted that they admit. Even Elon Musk, if he listened to this podcast, was saying all the time, we need to die. But nobody, when they are 80 and they have Alzheimer or Parkinson or they have diabetes, nobody would like, I think, to say, okay, I give up. If there is a treatment that allow me at the age of 80 still walk out in the street with my wife or my kids, people, I think, would like to do that. So we need to support more of these crazy out-of-the-box ideas and assume that if one of them work, it will make a huge change in our society, and a lot of people, a huge chunk of mankind that is able to access, this is a topic for another podcast, who can access, but a long chunk of mankind that can have access would like to use it. There is no other way, I think, to look into that, really. A lot of people that present in conferences or are investors or company heads, most of them are still young. Most of them are very coherent. Most of them are very active. These are not the people that, suffer currently, most of them, not all of them, that suffer from the progression of aging. But I bet when we all reach this end of the road or close to the end of the road, we would like to have the power to do something about it. I wish there would be more out-of-the-box ideas that can really help us as a whole group dedicated to treat human lifespan and longevity. There are more ideas that are supported, and we see what will be the potential.
Yes, fascinating. Well, kudos to you. It takes a lot of courage and curiosity and perseverance to handle a project like this, to have this out-of-box thinking and to actually apply it in real-life experiments and take it into translatability, start in a company, and truly moving the field forward. So I wishing you all the best. And I also wanted to build up on that point too. As you said, it could be applicable to so many other diseases. Like when we think about gene therapies classically and the way they've been developed in the beginning and where they're now and where the field is moving on to now is it started from monogenic disorders, the most debilitating one gene, one mutation, one disease kind of disorder. We can go and fix the gene or go and fix the mutation. Like now with base editing with CRISPR, we can, in a cleaner way to fix the mutation, you have to go and, of course, approach it from a way of knowing the specific disease population and natural history of that disease, and then adapting the technology specifically to be delivered in the cells that are most damaged, and so on and so forth. With technology that can be more applicable to a wider range of disorders, it's very likely that there's a lot of mutations that, again, impact the mitochondrial function to the extent that it impacts specifically the mechanisms of the energy homeostasis as maintained by mitochondria. So if instead of targeting any specific one mutation, especially mitochondrial mutations that are much harder to target, is it possible, if this is applicable in a way to replace, as you said, the energy mechanism inside of the cell, to actually target major, like most of these disorders? So I see it as having tremendous potential. And I am so grateful for having you, Shahaf, on our podcast today, because it's been an incredibly interesting topic. And thank you so much for coming on. Thank you so much for sharing your outstanding, outlandish thoughts with our audience. And I'm wishing all of the best to your project, to Luminova. Thanks so much.
Elena, I just want to say thank you very much for giving me the opportunity, and I wish you also great success in your podcast. You have enthusiastic subscriber. I will check also the next podcast, and really thank you very much for giving me the platform. Thank you very much.
Thank you for tuning in to the GTI podcast. I hope you enjoyed the episode and found new insights for yourself. If you like the episode, please press like, subscribe on your favorite platform. Your best support will be sharing this episode with colleagues and friends. For the future content, comment below with suggestions of guests to interview and questions you would like to be answered.